Hello friends, welcome to my channel Kempom. Today I will be explaining one experiment which you can present in open day anywhere, which is blue bottle experiment. This is one of the famous experiment to present in open days. In blue bottle experiment, the chemical required is methylene blue one as dye, and then you require glucose, NaOH, methanol and water. Here I didn't mention methanol and water, but you need methanol to dissolve methylene blue and make a dye, and glucose and NaOH in bulk you have to dissolve in water, and then few drops of dye you have to add. Here at first in conical flask or RV, at first you have to dissolve glucose, NaOH in water, and then few drops of methylene blue. Okay, then you shake for few minutes, and after that if you leave it, then that solution will be colorless. But if you shake that solution turns to blue. Then in static condition, if you leave that solution for few seconds or minutes, 30 seconds or 1 minute maximum, then again the solution becomes colorless. That means that blue color disappears. Again if you shake that solution, then that blue color reappear. This is the intro of blue bottle experiment. This is an interesting experiment to present in front of school students, like school students or kids. It's good to play and also not hazardous or anything, not dangerous. Simple experiment but interesting. And here the schematic diagram of the experiment. First, what happens that glucose oxidizes in presence of oxidized dye, that blue dye, and since that glucose oxidizes and hence that oxidized dye reduced, the reduced form of that dye is colorless due to lack of conjugation. But here that oxidized dye is blue in color. Okay, that's why at first while adding that dye and glucose solution, everything, that oxidized form of glucose forms and reduced dye, colorless reduced dye forms. That's why that one is colorless. But if you shake that solution, then in presence of oxygen in air, that reduced dye oxidizes to that blue form, and hence blue color reappear. Okay, and this one is the experimental demonstration details, like the mechanisms behind all this one, the NaOH, this form, this one is the functional part, that means participating functional group of that glucose moiety, and then NaOH, in presence of NaOH in presence of NaOH it forms this configuration, and then in presence of methylene blue it oxidizes, and then oxidized form it reduces, and in presence of oxygen that blue form it comes back to that blue form. This dye, in this form the dye is blue, in this form dye is colorless because of conjugation. If you look at this form, this one is oxidized form, then this one is aromatic system, this one is also in conjugation, and this system also in conjugation. That means 3 rings, 3 cyclic systems are in conjugation. Hence homolumo gap falls into visible region, and hence that blue color generates. On the other hand, this form of that dye, this ring and this ring, between this ring and this ring there is no conjugation. That means due to the presence of this single bond and this single bond here, this single bond, this single bond, these two systems are not in conjugation. Hence, homo-lumo energy gap increases, which doesn't fall into the visible region. Remember that in conjugation, alternative double bond, presence of alternative double bond is necessary. Okay. And next, that here I mentioned this slide because this slide actually one of the proof that in presence of oxygen blue color appears. That means oxidation, importance of oxygen to oxidize that reduced dye, because in air condition if you proceed this reaction then that blue color generate, but under nitrogen atmosphere, that means absence of oxygen, there is no formation of blue color, which confirms the importance of oxygen and also confirms the oxidation of that reduced dye to reappear the blue color. Thanks for watching. Please subscribe my channel for more videos. Thank you.